The last of my talks is on survival analysis, and this will be the most statistical of the talks. The other ones, were some interesting stories because, those are usually more interesting than statistics. But this is a technique that is often used in clinical trials, and people who aren't in clinical trials may not be as familiar with it. Um, so in survival analysis, what we're doing is we're looking at time to some event. The event could be death, or it could be the first event in a composite. So maybe you're looking at time to death or heart attack. So whatever came first, if you had a heart attack, um, then it's time to that heart attack. If you died, then it's time to that death. Um, so in the old days, what did people do? They compared the proportion of people who had an event by a fixed time, like 2 years, and they just compared the proportions. The problem with estimating the probability of having an event by a fixed time, 2 years, say, is that that time is often pretty arbitrary, because you're definitely interested in time in longer times as well, and maybe shorter times. Um, and the other problem is that in clinical trials, different patients have different amounts of follow-up time because of what's called staggered entry into clinical trials. That is, the patients are entering the trial at different times. Um, and sometimes patients move or they drop out of your trial, or maybe they die of some unrelated disease or unrelated cause, like a car accident. And so you have different amounts of follow-up in different patients. Now, if a patient drops out after 1 year of the trial, you don't know what would have happened in that second year. If you're using 2-year mortality, for example, you don't know what would have happened in that second year. All you know is that the time to death was at least 1 year. They hadn't died by the time they dropped out. So this is called a censored observation. And in particular, this is called a right censored observation because you don't know what happened to the right of 1 year, after 1 year, in other words. And so this picture, depicts what happens in clinical trials. You open the clinical trial, and let's say one patient comes in relatively early, and then they amass a certain amount of time, and then they have the event of interest. Let's say it's death, they die at this time. So their time to event is from when they entered and got randomized to when they had the event. Here's a patient, though, who arrived later in the trial after, say, 3/4 of a year, and they went to the end, or not the end, but let's say they dropped out of the study at this time. So we put a little plus sign to denote that this is a censored observation. They had not had the event yet up until this time, but then they dropped out of the study, so we don't know what happened afterward. Here's another patient who drops out. This last patient, has an event. You have patients entering at different times. You have patients, some of them are having events and some of them are not having events. Some of them are censored observations. Now, what we need to do in order to make progress in a situation like this is we have to make the assumption that the reason for not seeing the observation, in other words, the reason for the censoring, is unrelated to whether they were about to have the event, for example. This is so-called non-informative censoring. So examples where censoring is likely to be non-informative are when the participant moved because the company that they work for moved and they just went with the company and you lost track of them when they moved, or they didn't let you know that they were moving. Um, Another example is, you're looking at a certain disease and the participant gets killed by something that seems like a completely random event, lightning strike, say, or getting hit by a train, or something like that. And so that's really giving you no information about whether they were about to have a heart attack or not, if that's the outcome of interest. And then another example that's likely to be non-informative is so-called administrative censoring, namely the trial ended, and so of course you don't know what happened afterwards. They went to the end of the trial, hadn't had the event, and then the study ended. Here are some examples where the censoring is likely to be informative. Um, if the participant dropped out because they were getting worse, maybe they'll tell you that. Oh, I was just getting sicker and sicker, and I'm not going to stay in your study anymore. That's a problem, because that's a situation where the reason that they're dropping out is related to whether they were about to have the event. Maybe they were getting so sick that they would have had the event very soon afterwards. Um, uh, or a participant in a cardiovascular trial receives a heart transplant. This is kind of an interesting situation. You're looking at, say, time to a heart attack, and then the person gets a new heart. And so you feel like, oh, wait a second, that's not fair. I can't continue to look at time till they have a heart attack, because this is a new heart. That changes everything. But why were you getting a new heart? Obviously, you had to be very sick to get a new heart. So that probably tells you that you were more likely to have the cardiovascular event than you would otherwise. Or let's say you have a drug abuse trial and the participant is jailed for driving under the influence, and therefore couldn't attend their drug test. Well, obviously, if the reason is that they were jailed for driving under the influence, they probably would have failed that drug test. So these are all situations where the censoring is likely to be informative, although I was once the statistician in a clinical trial in which one of the MDs argued that this number two situation of receiving a heart transplant was actually not informative censoring in that particular trial, because he said all of the patients in this trial are so sick that they're all eligible to receive a heart transplant. The only difference is that this person got lucky and a heart became available. So it's actually completely random, and it's not informative censoring, which was very interesting. That gave a completely new perspective to me. All right, so let's look at an example of how you deal with these survival data. And the most popular method of plotting survival is called the Kaplan-Meier survival curve. So I'm going to illustrate how this works with a small example with only 11 patients. Don't do clinical trials with 11 patients or other studies with 11 patients. And let's suppose that 5 of the 11 have censored observations, and the rest, you actually have their time to the event. And so here are their times, and a plus sign denotes that that's a censored observation. If there's no plus sign, that means they had the event after that amount of follow-up time. So what we do is we can look at this first event time. We order them from smallest to largest. And let's look at this first event time. Now we have 11 patients in this study. And so how do we estimate the probability of surviving 0.8 years? Well, 10 of the 11 patients survived 0.8 years. Only one of them died, this one right here. And so we would estimate the probability of surviving 0.8 years to be 10 out of 11. Then what we can do is we can go to the next time, 1 year, and say, okay, what's the probability of surviving at least 1 year given that you've survived this 0.8 time? Well, 10 of them survived the 0.8 time, and all 10 of them survived at least 1 year, including the censored observation, because you know that they lasted this long and they had not had the event. So the probability of surviving 1 year, given that you survive 0.8 years, is estimated by 10 out of 10 or 100%. Likewise, we calculate the probability of surviving 1.2 years, given that you've survived at least 1 year. And there are now 9 patients remaining, and 8 out of 9 survived at least 1.2 years. And so we continue to calculate all these probabilities. Given that you survived the previous time point, what's the probability that you survive the next time point? And then what we do to get an overall estimate of the probability of surviving a certain amount of time, what we do is we say, well, the probability of surviving 0.8 years, first you have to survive 0 years. Everyone survived 0 years. Then you have to survive 0.8 years. And this was the probability of surviving 0.8, given that you survived 0. So we take 1 times 10 out of 11, and that gives us 10 out of 11. So you had to survive 0, and then given that you survived 0, you had to survive 0.8. So we multiply this probability times this probability, and that gives us about a 91% chance of surviving 0.8 years. Then to survive 1 year, you have to first survive 0.8, then you have to survive 1, given that you survived 0.8. So you take this 0.909 multiplied by the probability of surviving 1, given that you survived 0.8, which was 10 out of 10, and that also gives us 0.909. And likewise, we go zigzagging through this table, multiplying the probability of surviving the previous time point times the probability of surviving the next time, given that you survived the previous time. And that's how we do it. We just zigzag through the table, multiplying these probabilities. And this gives us our estimated survival probabilities. And then we make a plot. And this is the Kaplan-Meier survival plot. So the estimated probability of surviving is 100% all the way up until this first death time, which was at 0.8 years. And then the curve drops down, the survival probability drops down to about 0.91. Then it stays flat again until you get your next death, and then it drops, and it drops only at death times. There are some plus signs here that are very difficult to see. I should have made this a lot bigger. There's a couple of plus signs, or there's at least one plus sign. I think there's more than one, indicating a censored observation. And what you can see is that the survival probability does not change at a censored observation. It only changes at these death times. And then by the end of 2 years, your estimated probability of surviving is only 0.4 something, looks like it's 0.404. And so you get this nice picture showing you the survival. That's called the Kaplan-Meier survival probability. And we didn't have to make any assumptions about what kind of distribution this survival comes from. It's all what's called non-parametric, in the sense that we're not making any assumptions. Before, modern methods, people used to make assumptions about survival distributions. Now, it's common to see, rather than this survival plot, you will see 1 minus the survival plot. So you just see the plot of the probability of having an event by a certain time rather than not having an event by a certain time. So that's just 1 minus the survival probability. And so it goes up instead of down. And you often see, comparisons of 2 groups. So this is a vaccination with Allvac and AIDS vax in a clinical trial of HIV to prevent HIV in Thailand. And you can see that the group that got the vaccine seemed to have a little bit lower chance of getting HIV than the people in the placebo group. People in the placebo group had a higher chance of getting infected by a given time. The p-value is not striking though. It's 0.04. It's a little bit below 0.05. And I'll be showing you how you get that p-value in just a little while. But this is nice to see this graph where you see, the two groups, and you can see, visually that there's a better outcome in one of the groups. Okay, now things to note about this Kaplan-Meier curve is, as I mentioned, it steps down at each time there's an event, not at a censoring time. At the beginning of the study, you have a lot of people, and as you go out over time, you see fewer and fewer people. That's what this is showing here. The number of people is dropping over time because a lot of people haven't had a chance to reach 2 years of follow-up yet. And so the jumps in the curve are small at the beginning, and they're bigger jumps at the end. So you shouldn't take results later in the curve too seriously. The ones that have big jumps, you're seeing a lot more variability. And this is an example. Remember I mentioned the WAVE trial, the Women's Angiographic Vitamin and Estrogen trial, which was really looking at changes in angiogram results. It wasn't really looking at hard outcomes like heart attack or death. But they also did look at those other outcomes. So they did look at time to either death or a non-fatal MI, heart attack. And you could see, even in this trial, hormone replacement therapy versus placebo, it's kind of hard to see, but it shows there was an increased risk of death slash non-fatal heart attack in the group that got the hormone replacement therapy. So even in this trial, they were seeing an increased risk. But as I said, at the end of the trial, the point of this graph is to show you have these big jumps at the end of the trial. So this is unstable. You should not take whatever is happening over here too seriously. These little jumps are a lot more stable, and you can see that there's a clear separation between the two groups. Now, in the SMART trial in HIV, this was a trial that compared a drug conservation strategy to a viral suppression strategy. So the drug conservation strategy, you would give the people drugs and then see what their CD4 count was and T cell count, and see what their viral load was, the amount of virus in their system. And then if it was good, then they'd take you off the medication and say, okay, we think you can go off the medication for a while, but we'll continue to monitor your CD4 count and your viral load, and we may put you back on medication. And then the other group just remained on their medication the whole time. That was the viral suppression group. And you see that there's a big separation between the groups. The drug conservation group, this is the one where you go on and off the drugs as needed, had a much higher probability of having the outcome of interest, which was opportunistic disease or death from any cause. So a lot more diseases in the group that went on and off their drugs compared to the group that got continual drugs, the viral suppression group. And you can see that there's a low p-value of less than 0.001, big separation between the groups. And this showed very definitively that you should not go on and off drugs. You should stay on the drugs once you have HIV. And so this was a very definitive trial. And you even at the end of this trial, there's still quite a few people. And so these jumps at the end are still relatively small. And it's a good idea to put the number of people that have reached that amount of follow-up down here at the bottom of the curve, like we saw in these the two plots that I showed you. Now, whenever you're doing graphs, there's the there's the possibility of really distorting things by changing the scale or the start value. And here's an example from a cardiovascular and renal drug advisory committee that I was on the committee for this in 2005. Um, I wasn't on the committee, but I was a consultant, and I was at this advisory committee meeting. And they were evaluating the effect of everolimus, which is a drug to give patients who have had a heart transplant, and they were trying to, you know, make sure that they did not reject the heart. So this is a drug to help them not reject the heart. And this is, so the company, the sponsor, showed results from this clinical trial, and this was um, not having efficacy failure through 24 months. So the higher you are, the higher the curve is, the better in terms of, you know, you have a higher probability of not having this failure event. And what the company showed was this nice curve that showed that their 3 milligram dose did the best. Their 1.5 milligram dose was intermediate. And then the standard treatment, this is a different treatment, their standard treatment that they gave had the worst results. So very convincing, you know, dose response relationship, highest dose works best, lower dose still works better than the standard treatment. Now notice what they did here. They didn't start, they you know, have this little break things that show you that, okay, the distance between 0 and 40 is different than the distance between, you know, these other ones. 40, you know, each other one is only 10, but this one is 40, which is fine. They're telling you about it. They're calling your attention to it. So and the reason they're doing that is that so that you can see is the separation between the two groups. If you went 0 to 100 and you made each one 10, these three lines would sort of go together at the top, and it would be harder to see the difference. And I understand that, that's fine. But then later on, the same company showed this graph, survival through 24 months, Kaplan-Meier curve. And here it's very hard that, oh, here they didn't do that break. And so you see, you know, they're incrementing by 10. And so it's very much harder to see the separation between these curves. And if you could see the separation between the curves, you would see that the lower dose did the best, the other treatment did the next best, and then the highest dose actually did the worst. And you know, was this intentional? Did they you know, do this uh, this intentionally distort the graph so that you couldn't really see that difference? I don't know. I have my suspicions, but it's not a good idea to do this because the advisory committee was very skeptical about, you know, that presentation. So the next thing I wanted to talk about then is how you test for differences between survival curves. And the most common test is called the log rank test. And here's a small example. As I mentioned before, don't do these small clinical trials. This is just to illustrate how the procedure works. So let's say we have only 4 people in the control group and 4 people in the treatment group. And it does look like treatment might be doing better because you can see that 3 out of the 4 people in the treatment group made it 2 years and you know, were censored. They never did have the event. Only 1 person had the event, and they had it at 1.5 years. On the other hand, in the control group, 1 person had an event after only half a year. Another one had an event after 2 years. And then these other 2 people were censored observations. So there's some data that seems to suggest that maybe treatment works, but is this enough evidence? Well, the way we're going to test that is we're going to put the data together and order the data. And so I did that. I put all these numbers together and I ordered them from smallest to largest. And the blue ones are the ones that came from the treatment group, and the black are the ones that came from the control group. Then the next thing we want to do is look right before each event. So here's the first event after half a year. We're going to look right before that happens. And we're going to say, okay, look, we know that someone died right here at half a year. And what's the probability that that person, if let's suppose the treatment had absolutely no effect, then the probability that that person who died came from the treatment group is what? Well, there are 4 people in the treatment group, and there are 4 people in the control group, and it could have been any of those 8 people. So the probability that that first death came from a treated person should be 4 out of 8, or 1/2. Then we move to the next death. So we skip over this censored observation at 0.75, and we move over right before the next death at 1.5 years, and we say, okay, we know there's a death here at 1.5 years. What is the probability that that death came from the treatment group? Well, again, there are 4 people in the treatment group. It could have been any of those 4. And there are now there are 2 people in the control group that it could have been. And so the probability that that death came from the treatment group is 4 out of 6, or 2/3. And then we move to the next death time, which is 2 years, and we do the same thing. We say, given that there was a death, what's the probability that it came from the treatment group? Well, there are 3 people in the treatment group, 2 people in the control group. So if treatment has no effect, that probability that it came from the treatment group is 3 out of 5. So then we summarize our data. We looked at, okay, the first death, the second death, the third death. There were only 3 deaths in this trial. This is not a real trial. I'm making up this data, obviously. And so these are those probabilities that I just computed. The probability that it came from the treatment group, that death came from the treatment group. Here's the actual data in terms of whether it came from the treatment group or not. So we're recording a 0 if that death came from the control group, a 1 if that death came from the treatment group. And so the first and last ones came from the control group. The second one did come from the treatment group. And so now what we do is we add up these probabilities of coming from the treatment group. We get 1.767. And that tells us the expected number of people the expected number of deaths that we should see in the treatment group if treatment has no effect whatsoever. Then this right here, if we add up these 1s and 0s, this gives us the total number of treatment deaths that we actually observed. So this is how many we would expect to see if treatment had no effect. This is how many we actually saw. So now we say, okay, look, we saw, fewer deaths than what we expected, but is that strikingly fewer? Well, to figure that out, we take the O minus E, the observed number of deaths in the treatment group, minus the expected number of deaths, and then we standardize by dividing by its estimated standard deviation, and that turns it into a Z-score, which for a typical trial where we have a lot more events than this, we can use the standard normal table to figure out whether this is an unusually, unusually small Z-score or not. Now, in this trial, you really wouldn't be able to trust that. But as I say, in any reasonable clinical trial, you will have lots of events. And so you could refer this Z-score to a standard normal distribution to be able to compute the p-value. And that's what was done here for that trial that I showed you. This p-value right here was computed using the log rank test. Okay. Now, the log rank test is more powerful than just using a test of proportions. So one thing you could do, as I said, the thing that people used to do is they just calculated the proportion of people who had an event by some fixed time, like 2 years. This test, this log rank test, is more powerful, provided that the so-called proportional hazards assumption is met. Now, what does that mean? What's the proportional hazards assumption? What is the hazard rate? Um, so the way we define the hazard rate is it's sort of an instantaneous probability of having the event in the next tiny amount of time, given that you've already survived up to now. So it's the probability of having an event in the next tiny interval of time from now to now plus, from now to now plus a little bit, divided by um, uh, given. So that's a probability given you've survived up to now, and then we divide by the amount of time to turn it into a rate. So that turns it into a rate, and think of that as sort of an instantaneous risk of having the event. So, for example, if you saw 1 million people who survived 1 year after bypass surgery, if 12 of those died between 1 year and 1.01 years, that's a small amount of time, then we might estimate that hazard rate at 1 year to be 12 over 1 million. These million people survived 1 year, and then 12 of them died. So it'd be 12 over 1 million divided by the period of time, which is 0.01 years. And that gives us a hazard rate of 0.00112 per year, 1.2, sorry, per year. Now, the hazard rate could increase over time. For example, if you look at the death rate in 75-year-olds, 5 years later, they're going to be 80 years old and more things are failing. And so they're more likely to die in a small period of time when they're 80 years old than when they're 75 years old. So that hazard rate is increasing over time. On the other hand, the hazard rate and for other things might stay roughly the same over time. So for example, if you're looking at mortality in 30-year-old healthy people, these people are probably only going to die by some sort of catastrophe, a car accident, or something like that. Very few of them are going to die from some disease. So the probability that they die in a tiny interval time right now, um, uh, uh, is about the same as if you look 5 years later, because now they're only 35 years old. They're still probably dying from more or less random events. Um, and the example, I like to give an example. I used to be an engineering statistician for 1 year. And we looked at foreign object damage. That's where there was some damage to the engine caused by something something being sucked into the engine. Usually, this is a bird. That's what happened with the Sully Sullenberger incident over the Hudson River when he landed that plane over the Hudson. Um, that's what happened. Birds got sucked into the engine, and the plane, it destroyed the engine. Now, this is a more or less completely random event. It's a very rare event that birds get sucked into the engine, but it's completely random, and it's not more likely to happen with an older engine than a younger engine. It's just, it's just where the birds happen to be when you're flying through there. So the hazard rate there for having this engine failure due to foreign object damage is going to be about the same no matter how old the engine is. So that's a constant hazard rate. In another example, you might see a decreasing hazard rate. So for example, if you're looking at rejection of a new heart, the highest probability of rejecting a new heart is right after you get the transplant. If you've gone 5 years and you haven't rejected the heart, then there's very small probability that you're going to reject it at that point. So that's an example where the hazard rate is actually decreasing over time. Now, the hazard rate could increase for some part sometimes and decrease for other times and stay the same for others. We don't really know what the hazard rate is going to look like. This is illustrating the hazard function for Swedish families between 1751 and 1851, and they're looking at hazard for death. And you can see that there's a relatively high hazard very near birth, because of the healthcare back then. They couldn't prevent certain things that we can prevent now. Once you survive, a few years, then your hazard for dying is very low until you get much older, and then your hazard rate goes up and up and up. So, it could have this bathtub shape. It can have all kinds of different shapes. So what we don't want to have to do is to estimate what that hazard rate looks like over time. But now let's look at the ratio of hazard rates in the treatment group to the control group. So regardless of whether this hazard curve looks like, it might be the case that the ratio of treatment to control hazard rates is constant. That's called the proportional hazards assumption. We're assuming that the ratio of hazard rates in the treatment group to the control group is the same over different times. If the null hypothesis is true, the treatment doesn't have any effect whatsoever, then of course the proportional hazards assumption is true, because in that case, the hazard ratio should be 1. There should be no different hazard in the treatment group than the control group. On the other hand, if treatment is effective, then we would expect to see a hazard ratio less than 1, smaller hazard in the treatment group, but the hazard ratio might vary depending on time. But if we make the proportional hazards assumption, so here's an example where it varies by time. The hazard ratio might be 1/2, that is, we're reducing the hazard by 50% at 1 year, and 3/4, the hazard ratio is 3/4, so that means we're reducing it by 1/4 or 25% at 2 years, let's say. So this would be an example where we don't have proportional hazards. We don't have the same hazard ratio at 1 year versus 2 years. So the proportional hazards assumption is that we do have the same hazard ratio for all time. And if that is the case, if the proportional hazards assumption is met, then you can show that this log rank test is the best possible test. So that's great. That's what we usually do. We usually do the log rank test. In some situations, we don't have proportional hazards. So for example, there might be a treatment lag. It might take time to actually see a treatment effect. That was seen in this case of zoledronic acid, 5 milligrams, where they were looking at, that's a cholesterol reducing medication, I believe. And you'd see a 28% reduction in mortality, but it takes quite a while before you see that benefit kicking in. So, it took over a year before you started to see that separation between the curves. So this would be an example where you actually don't have proportional hazards. The treatment effect later on was much larger than early in the trial, where you seem to see no effect. So that would be an example where you don't have proportional hazards. You can also sometimes see survival functions crossing. That happened in the multi-center unsustained tachycardia trial, MUST. This is an interesting trial because this trial compared, again, antiarrhythmic medication to no antiarrhythmic medication for people who had cardiovascular issues. But in this particular case, the medication that they gave was based on using this electrophysiologic testing. And I don't know exactly how that works, but it's some kind of test that shows, okay, this person really needs to be on medication. This other person doesn't need to be on medication. And so they used that, and I was really worried in this clinical trial. I was involved in this trial because of, I was worried about what happened with the CAS trial. And I thought, why doesn't everyone expect the same thing that happened in the CAST trial? And they said, oh, well, we're using electrophysiologic testing. That's completely different. And so I thought, okay, maybe that's completely different. Um, well, it didn't seem to be completely different. So when we looked at the results, um, for people given the antiarrhythmic drugs, um, it seemed to be dangerous. It seemed to show some evidence of increased risk in the drug group. But the intervention was not just drugs, it was either drugs or an ICD, an implantable cardiac defibrillator. Those are little devices that you attach right to the heart, and they can detect arrhythmias and they can reverse them. Has the same effect as taking a defibrillator, putting it on the chest, and shocking your heart back into a normal rhythm. So early in the trial, it was the implantation of defibrillators was more difficult to do. Later in the trial, it became technologically easier. So they did more ICDs at the end of the trial. And then at the end of the trial, they actually got a statistically significant result showing a benefit of the treatment. But I believe that it was almost all the benefit of the treatment was because of the ICDs, not because of the drugs. And so they were trying to illustrate this, that it was the effect of the ICDs. So what they did was very clever, I thought. So they looked at they looked at uh, uh, they looked at before January 1st, 1994, results before January 1st to after January 1st, 1994. Now, the reason they're looking at that date is that is when it became much easier to implant the defibrillators. And so they actually changed the criteria for when you would receive a defibrillator because it became so much easier to do the actual surgery. They said, okay, now you don't have to fail 2 drugs before you get the defibrillator, even if you only fail one, or I should say the drug fails you, not you fail the drug. Um, but at any rate, so it became easier. That's why they split this into before it became easier to after it became easier. And you can see that, there's this hint of an early harm of the electrophysiologic guided therapy. And that's because they were mostly getting drugs. And then after January 1st, they're mostly getting the defibrillator, the implantable cardiac defibrillator, and you can see a big separation between the groups. So, so this is an example where you see non-proportional hazards before 1994, but it does seem to suggest an early harm of giving them these drugs. Now, the last thing I wanted to just talk about is the Cox model, which is a way to adjust for differences in other baseline covariates. So, maybe you want to see whether the groups really do have this difference in survival after adjusting for the fact that more people in the control group had, more serious disease or something like that. So you have some baseline measures of how sick they were at the beginning of the study, and you want to incorporate them into your analysis. You do that with the Cox proportional hazards model. It's very analogous to logistic regression, and you can include covariates that are binary. You can include continuous covariates. So it's really a way to just adjust for any baseline differences. Remember, in clinical trials, there shouldn't be big differences in baseline characteristics because randomization is deciding who gets into what group. But still, it's reassuring to see that after you adjust for any baseline differences, you see similar results, which is usually the case. So just in summary then, survival methods are useful whenever you're looking at time to an event. The Kaplan-Meier curve is a very useful curve to to compare the groups. And the log rank test is a very useful method for testing whether there are differences between the groups. And these methods, both of these methods, and most of the survival methods are really only handle non-informatively censored data. And if you do have informatively censored data, like a serious amount of informatively censored data, then you have to consider other methods like sensitivity analyses, where you say, okay, let me count this person as if they died at that time and see if that changes the results. So these methods that I've shown you are not valid if the patient quit because they were getting worse, for example. And then hazard rate is like an instantaneous event rate in the next tiny interval given that you survived up to now. The hazard ratio is the ratio of those rates between the treatment and the control group. And that's really analogous to like a relative risk. And the proportional hazards assumption means that you're assuming that that hazard ratio is the same for all time. And if that's the case, then the log rank test is the best test to use. And you can also adjust for any baseline covariates using the Cox proportional hazards model. So I will stop right there. It's just a summary of a lot of different things: continuous, binary outcomes, and survival outcomes. But what I was talking about today was the survival outcomes. So I will stop there. Thank you.